We're well aware that endurance exercise is good for us, but have you ever wondered how it's good for us? What mechanisms are triggered by endurance training to benefit our health? Fortunately, there's a large group of researchers who have come together to tackle this question and assess the wide-ranging impact that physical activity has throughout our body. This group, called MoTrPAC, Molecular Transducers of Physical Activity Consortium, is planning multiple phase studies to look at the immediate or acute effects of exercise, both endurance and resistance, as well as the sustained effects of endurance and resistance training, first in rats and then in adults and kids. The reason to use rats as an initial model is so the researchers can probe for tens of thousands of biomolecules from various tissues and organs in a way that can't feasibly or ethically be done in humans. So this first step cast a wide net to determine what molecular elements are involved during a course of endurance training. And the results, which identified over 35,000 factors, were published on May 1st in the journal Nature. I won't dive too deeply into the details of the study because, to be perfectly honest, I can't. This type of science is more complicated than what I can wrap my head around, and I struggle just to pronounce the names of some of these molecules. But I do understand the gist of what they're trying to accomplish and the enormous potential that this work has to advance our understanding of how activity changes our bodies for the better. So I want to paint in broad strokes what they found by showing a few key figures from the article. Here's the title: Temporal Dynamics of Multi-omic Response to Endurance Exercise Training. The suffix ome, O-M-E, or omics refers to the totality or collective grouping of components. For example, while you may have a study that looks at the effects of a single gene, genomics takes into account the overall genetic makeup of an organism, so in this case, a rat. And the impressive thing about this project is that they're not just looking into the genome of the rat, they're looking into other omics, like proteomics, the full array of proteins that are involved from endurance training, along with omes that influence gene expression, the immune system, and metabolism. The gap in understanding that the consortium is trying to bridge is that in the past, most exercise studies are confined to only one type of molecular effect, or they may be limited to only one or two tissue types, like skeletal muscle or blood, and often they've only been conducted in males, with the assumption that the effects are the same for females. And it's understandable that prior studies have been like this, because to do quality research is expensive and it's time-consuming. So trying to prove or disprove a specific hypothesis is easier and actually more accurate the more focused the study is in limiting the variables. And that's what makes this MoTrPAC project even more remarkable, because their goal is to be comprehensive and look at multiple different omic categories from multiple organs and tissues. And these are shown here: the brain, heart, lung, liver, colon, small intestine, kidneys, adrenal glands, testes and ovaries, 2 muscle types from gastrocnemius and vastus lateralis, blood, as well as both types of adipose tissue, brown and white fat. The protocol from this first study involved comparing an inactive control group of male and female rats to groups that ran on a treadmill with steadily increasing incline, duration, and speed over an 8-week period. A subset of the animals were sacrificed after 1, 2, and 4 weeks, and the final group after 8 weeks to assess how the changes evolved with cumulative training. A quick timeout here. Yes, I have pets. Yes, I find sacrificing animals for science a difficult dilemma. I've had to do it myself once. And I'm also very interested in the information that we can only get from studies like this. This illustration shows the number of statistically significant training-regulated features that they found according to omic category, represented by the colors on the left column. So orange are factors associated with the genome in the top 3 rows, proteins in the next 4 rows, the immune system in pink, and metabolism in green. The vertical columns correspond to the different organ or tissue types, and the color of each cell relates to the proportion of measures that were significant based on the key here to the right. So a yellow cell indicates that of all the biomolecules measured in a given system, 10% or more were statistically significant compared to the control. As an example, if we look at the genomic changes in white and brown adipose tissue, nearly 3,400 transcription-related factors were altered in the course of the 8-week endurance training. That's mind-blowing. This is an illustration from the paper representing regulatory enzyme activity and signaling pathways that were affected by endurance training. Again, without diving into the details, what's striking here is the sheer number of factors, each represented by a dot on the grid, that were either enriched, shown in shades of red, or diminished, represented in shades of blue, as a result of the 8-week training. The size of the dots positively correlates with the degree of statistical significance. And at the top, you see the different organs or tissue types. The sex of the rats is denoted by pink or blue, and the yellow, green, and black columns represent the 1, 2, 4, and 8-week time points of testing. Every dot on this grid represents a molecular messenger that communicates within or between cells at the local level or remotely, sending signals out to other areas of the body. And previous studies have shown that there's interaction between pathways, which allows for crosstalk between tissues and organs. Take a look again to get a sense of how much internal communication is happening. Although this study is looking at endurance training, this systemic interaction has been shown in other forms of exercise as well. So while you're doing a biceps curl, the muscles may be sending out a healthy hello to your liver or kidneys. Lastly, here's a figure depicting the response for immune modulating factors separated by sex difference, again with pink for females, blue for males, and gray for when there was a similar effect in both sexes. For example, in the small intestine, with the exception of one factor, there is either shared similarity or a much more pronounced effect for the females. The opposite was true for the immune modulating activity in the brown adipose tissue, lung, and adrenal glands, which had several shared factors, but a more predominant and diffuse effect in males. In white adipose tissue, there were opposite responses for 2 factors between females and males. The researchers point out that this clearly demonstrates that results of single-sex exercise studies cannot automatically be extrapolated to the other sex. And in the future, studies will need to be formulated differently to test males and females. To ensure that their data has value that can be translated to humans, the researchers integrated their results with existing exercise studies and found multiple areas of significant overlap in transcriptomic, proteomic, and genomic investigations, which they state confirms a high correlation between their data from rats and what's currently known in humans and human disease. This study is a first step in trying to build a comprehensive map of the molecular response to exercise. So it's not one from which the researchers tried to draw firm conclusions. Instead, they present it as a foundation to continue their work and a resource that they are inviting other investigators to use to generate their own questions and launch their own projects. To support this, they've made the tissue samples accessible to other researchers, and the data sets are publicly available on their website, which is linked in the description, along with a full text link to the article. The MoTrPAC project is slated for 10 years, and I can't wait to see what discoveries they have in store. Even though the researchers didn't offer any conclusions, my takeaway from this is something that we all know but often forget. Our bodies are amazing. Next time I'm struggling on a run, I'll take a moment to imagine the 35,000 plus molecules streaming through my system, working to keep me healthy. Let me know in the comments if this is something you've tried to keep you going during endurance training. In case you missed it, here's a link to last week's premiere episode of Healthy Aging Headlines. Thanks for watching. I'll see you soon.